Good morning, my dear students. So today we are going to be learning about a molecular diagnosis under biological applications in medicines. In this molecular diagnosis, as you know, the diagnosis is nothing but detecting the disease. So here, many of the diseases were there. If they are going to be detected at an earlier stage, they can be get cured. So the detection are of two different types, like one a molecular basis and another a conventional method. So we will go for learn what is this conventional method and what is this molecular basis. But first you must understand the primary, secondary and tertiary stages of the disease. So if certain diseases are like a cancer, AIDS, which have not any medicines yet today, but still they can be get cured if they have been diagnosed at a primary stage. Say for example, cancer is there, so like a colon cancer or liver cancer, if it is a initial stage, you can cut the liver and throw it out. So at least you can save the life. So in this way, primary diagnosis. So if it is a secondary and a tertiary, nobody can do, just we can drag the life. This is the important. So under this molecular diagnosis, the conventional method, two conventional methods. The one said to be as a serum and another urine test. So serum is nothing but taking the blood, observing its contents present in the blood itself. Say for example, malaria is there. Malaria causing causative pathogen, as you know very well that when it enters inside the body, it is going to be multiply in the blood cells, then it will enter into the liver, it is going to become multiply like anything. After multiplication, it will start showing the symptoms. After symptoms, we are going for checking, just to find out whether this is present or not. Then the doctor will give the medicine, the person will become cured. So that is the drawback of serum test. So conventional means already the infective stages become incubated. Incubation period ದಲ್ಲಿ ದಾಟಿ ಹೋಗಿರುತ್ತೆ, ಅದಕ್ಕೆ ಅವುಗಳು ಗುಣಲಕ್ಷಣಗಳನ್ನು ತೋರಿಸಿದ ಮೇಲ್ಗಡೆ ಫೈಂಡ್ ಔಟ್ ಮಾಡುವಂತಹ ಮೆಥಡ್ಸ್ ಗಳಿವೆ. So there is no use. Like urine test also. Understand? Say for example, diabetes, in the first PUC you have been studied the glucose test. The urine sample test is going to get converted into brick red, which is showing the presence of glucose. So after infection of the disease itself, so conventional methods are going to be just to find out the disease, but here in the molecular basis, to find out in the earlier stage and get cure actually. So what those two diseases are, the one, those two diagnostic, not diseases, to find out the diagnostics, to find out the disease, to diagnose. These are the two. One is PCR, that is polymerase chain reaction, and another one is ELISA, enzyme linked immunosorbent assay. So polymerase chain reaction and immunosorbent assay, enzyme linked immunosorbent assay are two important molecular diagnostics used for diagnosing the disease under biotechnological applications in medicines. So as you know, what do you mean by this PCR and ELISA, right? So this is about molecular diagnosis. Then the second part of this is transgenic animals. So in this transgenic animals, the animals which have been manipulating the different genes which the product R is in need. The desired product can be obtained by introducing into the animals, and those animals are called as transgenic animals. Manipulation ಅಂತ ಕಲಬೆರಕೆ ಮಾಡೋದು. ಇವಾಗ ಒಂದು ಹಸು ಇದೆ, ಹಸು ಆಕಳು, ಆ ಆಕಳು ತನ್ನ ಪ್ರಕಾರ ತನ್ನ ಪ್ರೋಟೀನ್ ಅನ್ನು ಕೊಡ್ತಾ ಇರುತ್ತೆ, ಇಟ್ಸ್ ಅಕ್ಸೆಪ್ಟಬಲ್, ಇಟ್ಸ್ ಅ ನ್ಯಾಚುರಲ್. ಆ ಪ್ರೋಟೀನ್ ಅನ್ನು ತೆಗೆದು ಮನುಷ್ಯನಿಗೆ ಬೇಕಾದಂತಹ ಪ್ರೋಟೀನ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವ ಜೀನ್ ಅನ್ನ ಆಕಳ ಒಳಗಡೆ ಹಾಕಿದ್ರೆ ಅವಾಗ ಆಕಳಿಗೆ ಏನಂತ ಕರೀತಾವೆ ನಾವು? ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಎನಿಮಲ್. Understand? So the animal which got manipulated by the different gene to get the desired product are called as transgenic animals. The question is that why we need these transgenic animals? What is the importance of that? What may be the reasons why we have to be produce the transgenic animals? So these are the 5 important points. Most important question for 5 mark, 3 mark like that is the one among that is normal physiology in the development. Say normal physiology. Many of the people will not have a normal physiology. Someone will be short, someone will be more tall, someone will be very fat, someone is very thin, someone digestive system is not properly working, someone is not getting hungry, someone hairs are not growing. These are physiological defects, improper growth. Improper growth of any organ. It may be hand, it may be toes, it may be nails, hairs, nose, anything. So that is normal physiology and development. ಸೊ ಅಂದ್ರೆ ಏನು ನಾರ್ಮಲ್ ಫಿಸಿಯಾಲಜಿ ಮತ್ತು ಡೆವಲಪ್ ಆಗ್ತದೆ ಏನಿಲ್ಲ. ಇವಾಗ ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ರಿ, ಒಂದು ಜೀನನ್ನ ತಗೊಂಡು ಇನ್ನೊಂದು ಎನಿಮಲ್ ಒಳಗಡೆ ಹಾಕಿ ಅದನ್ನ ಡೈರೆಕ್ಟ್ ಆಗಿ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ ಒಳಗಡೆ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದರೆ ನಾರ್ಮಲ್ ಫಿಸಿಯಾಲಜಿ ಚೇಂಜ್ ಆಗುತ್ತಾ ಏನಂತ ಗೊತ್ತಾಗಲ್ಲ. ಸೊ ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಇಂಟ್ರೊಡ್ಯೂಸಿಂಗ್ ಫಸ್ಟ್ ಇಂಟು ದಿಸ್ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಎನಿಮಲ್ಸ್. ದಟ್ ಈಸ್ ಇಟ್ಸ್ ಇಂಪಾರ್ಟೆನ್ಸ್, ರೈಟ್? Whereas study of diseases, second one is study of disease. Say for example, ಇವಾಗ imagine ಮಾಡಿ, as you know very well that ಇವಾಗ COVID-19 is going on. COVID-19 virus. Do you think the medicine which are going to be discovering are directly injecting into the human being? If that medicine prepared is, can we inject into you? You say no, I will not. Why? Because you are acting as an animal. Which experimental animal. So they want to introduce first it into the laboratory animal, and those laboratory animals are the transgenic animals itself, because they must have almost all human physiology one. So that is the one study of disease. Whereas third one is biological products are going to be produced through these transgenic animals. Biological product. So one example I would like to tell you here is that alpha-1 antitrypsin. Alpha-1 antitrypsin is a protein, is an enzyme which is going to be curing a disease called as emphysema. Emphysema is nothing but respiratory disorder. Painful breathing. Painful breathing is due to the deposition of phlegm around the alveoli. So that is said to be as emphysema. This emphysema is treated by a protein called as alpha-1 antitrypsin, and this alpha-1 antitrypsin is produced through the help of a transgenic animal by injecting that particular gene into it. So many of the people are going to get cured from this. That is the one. Whereas in the year 1997, most important for 1 month, transgenic cow, the name of that cow is Rosie, and that is going to be producing alpha-lactalbumin. What is this alpha-lactalbumin? Alpha-lactalbumin is a protein present in the milk of the mother. Many of the mother which gives birth to the young one are unable to produce their milk through the help of the breast. They cannot feed the breast. When the babies cannot feed the breast,